Today we will be doing respiration in plants. The first lecture in this series. We will start. Now, first we have to know what is respiration. Now, respiration, if I ask the students, then they will simply say it is the inhalation and exhalation taking place in living organisms. That is inhalation of oxygen and exhalation of carbon dioxide. Now, beyond that also we have to understand respiration. Respiration is the process whereby there is evolution or release of energy for carrying out all our metabolic activities. So let us start with it. Let us try to understand how actually the respiration process takes place in organisms, and more specifically here we will be learning how it takes place in plants. Now, respiration. Okay, what happens in respiration? So first we have to know that is respiration. So respiration is what? It is the breaking down of carbon-carbon bonds. Now, we have learned about photosynthesis, or we know about photosynthesis, that it is the process whereby there is production of glucose, which are carbon molecules. So when those carbon molecules are again broken down, the bond between the carbon existing, that is broken down, that will release energy, and that energy release is in the form of heat. That is, the release of energy is respiration, and the process of that breaking down is called as respiration. So now here you can see light energy is absorbed through the process of photosynthesis. It is absorbed by plants, and there is production of glucose. Okay, glucose is produced. Now, during that process, what happens? Carbon dioxide is taken in. Okay, carbon dioxide is taken in, and there is production of glucose. There is the output of glucose. This white line, it indicates, the white arrow indicates the light energy, how it is utilized. Light energy is utilized by carbon dioxide in the form of ATP, and then finally there is release of glucose. Now what happens? The glucose that is present in plants will be broken down. Okay, will be broken down during the process of respiration to release carbon dioxide, and for that breaking down, what is required? Oxygen is required. So with the help of oxygen, what will happen? The glucose, it will be broken down finally to release carbon dioxide and energy in the form of ATP. That is what is basically respiration. Now this breaking down process, it's not so very simple. Okay, it involves a series of reactions. Now we have to understand those series of reactions. Now, respiration. Again, the definition. This is a process in which complex organic substances like carbohydrates, proteins, fats are broken down to release carbon dioxide and water. Now, carbohydrates, that is glucose, and which is broken down as I showed you in the previous slide. Even proteins present in the living systems can be broken down to release energy, or fats can also be broken down to release energy along with carbon dioxide, which will be the byproduct, and water. So that entire process is called as respiration. Now, as I told you, it can be represented. That means it is the breaking down of carbon bonds. So here it is C6H12O6, which is glucose. It reacts with oxygen to give carbon dioxide and water along with energy. Now this is 686 kilocalories. That is energy that is released per glucose molecule that is broken down. So that is respiration. So breaking down of the carbon bonds that exist between the 6 carbon molecules of glucose. That will be what we will be talking about now. Now what it requires? It requires a substrate, and this substrate, if you want to call it specifically, it is called as respiratory substrate. Okay, so substrate is required, oxygen is required, and then the enzymes, of course, it is fully enzymatic. Enzymes will be required, and it takes place inside the cells. Okay, specific locations inside the cell, mainly the involvement of cytoplasm and mitochondria. So here the respiratory substrate may be carbohydrate, may be fat, and sometimes it may be proteins. Mainly these are the three types of respiratory substrates. Now what happens basically? Okay, the outline of respiration I'll show you here. Glucose molecules present in the living system, which are 6 carbon molecules, okay, in the cytoplasm of the cells. In the cytoplasm of the cells, there takes place a series of reactions, fully enzymatic, which we call as glycolysis. Now always you should remember lysis means splitting. So here splitting of glucose molecules in 10 steps. 10 steps it takes place to give us a molecule that is pyruvic acid or pyruvate, which is a 3 carbon molecule. 3 carbon molecule, that is, so 2 molecules of pyruvate is yielded from each glucose molecule. Glucose, which is 6 carbon, it will yield 2 3 carbon molecules, each of them called as pyruvic acid, plus some amount of energy, minimal amount, but it is released. And where does it take place? In the cytoplasm of the cells. After that, the pyruvic acid that is present, now it will depend upon the presence or absence of oxygen. What will happen to this pyruvic acid? If suppose, okay, in animals, here in animals or even in plants, okay, in animals as well as in plants, if there is lack of oxygen, oxygen is deficient in that particular cell, then what will happen? The pyruvic acid will get converted to lactic acid and release energy. But the amount of energy will be very, very less. And if it is in a microorganism, this event is happening, the pyruvic acid was formed in a microorganism, there is lack of oxygen, then it will undergo fermentation, it will release ethyl alcohol and energy. Okay, it will release ethyl alcohol and energy. But here we are mainly talking about aerobic respiration. Now these two can also be called as anaerobic respiration. The aerobic respiration, that is presence of oxygen, what will happen? The pyruvic acid, that means it is coming in contact with oxygen, sufficiently high oxygen, then in the cells, then what will happen? Reaction will take place in the mitochondria of that particular cell where glycolysis took place and release carbon dioxide, water, and sufficiently high quantity of energy. Sufficiently high quantity of energy, and this is what is known as the aerobic respiration. Also it can be called as Krebs cycle. So these are the three events that can happen within the cell depending upon the presence or absence of oxygen. Now glycolysis. Okay, we will now further go into depth. Glucose. Okay, so glucose, the first, always when respiration starts, it starts with glucose. So that is known as glycolysis. Where does it occur? In the cytoplasm of the cells. Now glucose is partially oxidized into 2 molecules of pyruvic acid. So 2 molecules of pyruvic acid, as I told you, then it may either undergo aerobic or anaerobic. If it is aerobic, then glycolysis, immediately after that means there is oxygen, sufficiently high quantity of oxygen, then glycolysis will be followed by, that means oxygen will react and then it will go to form, it will go to carry out citric acid cycle or Krebs cycle. Okay, that will yield certain products. Okay, it will yield certain products. It's a cycle. Okay, it's a cyclic manner it takes place, and after that it will go to electron transport chain. So whatever products are yielded here, mainly NADH, okay, mainly NADH, FADH2 are the products that will be yielded here. So those products, what will happen? They will undergo another chain which is known as electron transport chain. That electron transport chain will finally give us the energy that we are talking about, will finally give us ATP, and both these events will take place in the mitochondria. This first step, it will take place in the matrix of mitochondria, and the second step will take place in the within the membrane, the inner membrane of the mitochondria. So finally there will be yield of ATP. That is why mitochondria is called as powerhouse of the cell. But if oxygen is absent, then what will happen? The glycolysis, after glycolysis, the pyruvic acid will undergo fermentation to yield alcohol or organic acid like lactic acid, and that will happen in the cytoplasm. That means it will end up in the cytoplasm. No event will take place in mitochondria if there is absence of oxygen. So this is the outline. Now we have to go into details. First, today we will start with glycolysis. Now glycolysis. Okay, glycolysis, what happens? Where is it taking place? It takes place in the cytoplasm. So we are assuming that the cytoplasm has glucose, has a good quantity of glucose. So that glucose, what will happen to it? It will first get phosphorylated. We call it phosphorylation. Phosphorylation means addition of phosphate. Now the glucose molecule, okay, at the sixth position, it is a 6-carbon compound. At the sixth position of carbon, what will happen? It will take up a phosphate molecule. From where will it get the phosphate? It will get from ATP. Now, ironically, what do we see here? We are talking about respiration, which gives us energy. But at the very first step here, energy is utilized. Okay, energy is utilized. ATP will be utilized and it will give out its phosphate. It will break down to form ATP, triphosphate to diphosphate, and one phosphate will be taken up by glucose to form a compound called as glucose 6-phosphate. This will be helped by the enzyme hexokinase. After which, the glucose 6-phosphate will get isomerized. Isomerization. I think you know what is isomerization. I suppose isomerization is just the structure changes, but the composition remains the same. So what will happen here? It will reorient. Isomerization will take place with the help of the enzyme isomerase, phosphohexose isomerase, to form fructose 6-phosphate, which was glucose, will now form fructose 6-phosphate. Then followed by it, what will happen? There will be addition of one more ATP, utilization of one more ATP, thereby one more phosphate molecule is getting added. Even this is a phosphorylation reaction. So in this, what will happen? What will happen? Phosphorylation means the addition of phosphate. So which was fructose 6-phosphate? That means only one phosphate molecule was present at the sixth position. Now one more phosphate will get added to the first position of carbon, thereby making it fructose 1,6-biphosphate. Now a very important reaction takes place here, and a very significant one, that is splitting. Splitting reaction. Now in splitting reaction, what will happen? This fructose 1,6-biphosphate, it splits. Okay, it will split into two compound, that is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Okay, or phosphoglyceraldehyde. It means the same, phosphoglyceraldehyde and dihydroxyacetone phosphate. So it will split into two compounds. Now two molecules. Now this dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, these are actually again isomeric compounds. That means the structure wise, formula wise or composition wise they are the same, but structure wise they are different. Okay, so that is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate is formed as a result of splitting of fructose 1,6-biphosphate. Okay, so here now what happens? This is a very significant step. Why I'm telling you, you have to understand it very well. Now what will happen here is glyceraldehyde 3-phosphate will continue with a series of reactions which I will show. Simultaneously, dihydroxyacetone phosphate will also continue with a series of reactions. Okay, both the series of reactions will be similar. Okay, they will be carrying out the same set of reactions. Parallelly, two sets of reactions will take place. So whatever yield we get after this, okay, it will always be in double. So if glyceraldehyde 3-phosphate forms the next compound, even dihydroxyacetone phosphate, first it will get converted to glyceraldehyde 3-phosphate and then it will continue with the same formation of the same compound. So whatever it forms, it will be in sets of 2. Now, what will happen? Glyceraldehyde 3-phosphate will get converted to 1,3-biphosphoglycerate. Now here, I think you have already understood that fructose 1,6-biphosphate was a 6-carbon compound. It's splitted, so 3 carbon and 3 carbon. Already the 3 carbon compound has been formed. Now with the 3 carbon compound only, reactions will occur here. Now here what will happen? Glyceraldehyde 3-phosphate dehydrogenase enzyme will act to form, to convert glyceraldehyde 3-phosphate to 1,3-biphosphoglycerate. 1,3-biphosphoglycerate. And here there will be yield of 1 NADH. There will be yield of 1 NADH. Then next, 1,3-biphosphoglycerate will also get converted to, now what will happen here? There were 2 phosphates, biphospho, 2 at the first and third position of carbon. Now 1 phosphate gets removed. Dephosphorylation. There is removal of 1 phosphate from here, and that phosphate will be added up to 1 ADP molecule, thereby forming 1 ATP molecule directly. 1 ATP formation takes place here. 1 molecule of ATP forms here because 1 phosphate got removed and it got converted to 3-phosphoglycerate or glyceric acid. Next, this is simply structural isomerism. So positional isomerization. So what will happen here? 3-phosphoglycerate will get converted to 2-phosphoglycerate. That means phosphate was the third position of the carbon. Now it will go to the second carbon, just shifting. Then this 2-phosphoglyceric acid will undergo dehydration reaction. That is H2O will be removed and it will form phosphoenol pyruvate, a compound called phosphoenol pyruvate. Now the last step, final step, will be formation of pyruvate. So here what will happen? There will be again phosphoenol pyruvate. You can see there was 1 phosphate group. This phosphate was present. This phosphate will also be removed to be added up to 1 ATP molecule to form ATP and thereby finally release pyruvic acid or pyruvate. So this is how pyruvic acid will be formed here. Now this pyruvic acid, now this pyruvic acid that is formed, as you can see here, we are showing on the one line here, glyceraldehyde 3-phosphate getting converted one after another to finally form 1 pyruvic acid molecule. Now this pyruvic acid molecule, that is formed here, this is 1 molecule. As I already told you, the dihydroxyacetone phosphate will also undergo similar type of reactions. Similar reactions will occur. So even through this line, you will get 1 pyruvic acid. Even here, phosphoenol pyruvate, here 2-phosphoglycerate, 3-phosphoglycerate, 1,3-biphosphoglycerate will be formed. So along with that, there will be release of NADH even from dihydroxyacetone phosphate. There will be release of ATP, there will be release of water, there will be release of another ATP molecule. So from here, you can multiply everything into 2. We just simply multiply by 2. So everything here, it is in double. Why we are saying double? I repeat, because parallelly 2 sets of reactions are occurring. So finally, there is yield of 2 pyruvic acid molecules. That is the end product of glycolysis. So finally, what do we get? We get 2 NADH molecules and here 2 ATPs. 2 into 2, 4 ATPs, but there is utilization of 2 ATPs. So 4 minus 2, we get the final yield will be 2 ATP, 2 NADH. The final yield of glycolysis. Along with it, with the formation of 2 pyruvic acid molecules. This pyruvic acid molecule, if there is presence of oxygen, then it will again further it will undergo to form lactate. It will carry out the reactions, but it will depend upon the presence or absence of oxygen. Now here again glucose, here it's glycolysis taking place, where there was utilization of 2 ATPs. These 2 ATPs glycolysis to form pyruvic acid molecule. So to form pyruvic acid molecule, and this pyruvic acid pyruvates as two molecules of pyruvic acid. Each of them, if oxygen is not present, will undergo fermentation to form either an organic acid, that is lactic acid, or ethanol. And if oxygen is present, then it will undergo oxidative decarboxylation, Krebs cycle, electron transport chain, and finally yield up to 36 ATP molecules from one glucose molecule. And these events will take place inside the mitochondria. So, where does glycolysis take place? Inside the cytoplasm. Once pyruvic acid is formed, the pyruvic acid will enter into the mitochondria, and in mitochondria, it will following these reactions will take place in the mitochondria. So that was all about glycolysis. Next, we will talk about this fermentation and oxidative decarboxylation.